Hello, everyone, and welcome to today's webinar, Development Advice for CBD Products, presented by Rowe. I'm Joe Keenan, contributing editor to Fierce Markets, and I'll be moderating this webinar. Our speakers today are Jack Modell, MD, Vice President and Senior Medical Officer at Rowe, Catherine Marr, PhD, Vice President of Regulatory Affairs and Quality Assurance at Evofem, Scott Burian, PhD, Senior Research Scientist at Rowe, and Dana Minnick, PhD, and a Senior Research Scientist at Rowe as well. You can read their full bios on the left side of your window by selecting the Profile button on the bottom of your screen. Just a few technical notes before we begin. If you'd like to download the slide deck, please click the orange Handouts button at the bottom of your screen. The webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. This webinar is being recorded and will be available on demand within 24 hours after the event. Time permitting, we will follow the presentations with a Q&A session. Please submit your questions using the Questions and Answers tab on the left side of your screen, and you can do that throughout the presentation. All right, we're ready to begin. Jack, please go ahead.
Great, thank you very much. Welcome everybody, it's a pleasure to be here. We've got quite a few things to go over with you today. We'll talk a bit about the history of cannabis, some interesting aspects about development of cannabis products from a clinical, regulatory, and also CMC standpoint. And then at the end, as mentioned, we should have time for specific questions. So just beginning now, going back to the history of cannabis, I think many of you know that this is one of the oldest known medicinal plants, actually going back about 3,000 years now, starting in Asia and Africa, and eventually migrating on to the Middle East. It hit Europe about 2,500 years ago, actually, and began to be used quite a bit around that time. But then on and off during the course of history, as we've even seen over the last few decades, appreciation of the potential medicinal qualities of cannabis has waxed and waned. But eventually, again, by the mid-1800s, 1840, you see here, therapeutic use was introduced in Europe. And for many years, about 70 years thereafter, it increased in popularity in Europe and the United States for all sorts of indications, menstrual cramps, asthma, cough, insomnia, labor pains during birth, migraine, throat infection, withdrawal from opium use. We know today that some of these indications, cannabis and cannabinoid products may actually help. Probably didn't help others, such as throat infection. But, you know, like a lot of things early on, it was tried for just about anything that might ail somebody and anything they could sell it for. But fortunately, over the years, we've learned more and more just what the cannabinoids can be helpful for and where their use is probably not as valuable. There have been dozens of cannabis and cannabinoid preparations available over the years. The problem is, and here's one actually from the Motor City back in, I can't quite read, I think 1924, Park Davis actually manufactured and sold an ethanolic cannabis mixture. The problem was that maybe not so much with Park Davis, but many of the suppliers, there were several, and there was lack of quality control, lack of standardized preparation. So dosing was inconsistent, efficacy therefore also inconsistent. Not dissimilar from what we see today in the unregulated products, the CBD products that you see pretty much everywhere you look now. Those are also unregulated and in some cases contain actually no CBD oil, and in other cases contain more THC than they actually should by law, and we'll talk about that a bit more in a minute. In the 1930s in the U.S., cannabinoids started to become vilified and blamed for all sorts of societal evils, to the point where by the 1950s, you pretty much have seen a disappearance of cannabis from Western pharmacopoeias. And in the late 1950s, the World Health Organization declared that cannabis no longer serves any useful medical purpose. That was based probably more on politics and experience with the inconsistent cannabis products that were on the market than actual scientific research, but that was the position from the 1950s for about the next 30 years or so. The raw materials, I think most of you are familiar where this comes from. The cannabis plant, like most plants, is made up of hundreds of organic chemical compounds. And in terms of cannabis, the cannabinoids and the terpenes in the cannabis plant, and the terpenes are the, best described as the aromatic compounds, which have smells and tastes that are very distinctive. They may too have their own therapeutic activities. These are produced in the plant's resin glands, which you can see there near the top, called trichomes. And the largest concentration of trichomes are found in the flowering heads of the female plant. So here you see actually a close-up of the glandular trichomes, which you see there's pretty significant concentrations and quantities of the terpenes and the cannabinoids from this portion of the plant. So cannabinoids, as I think many of you know, it's one of a class of diverse chemical compounds that acts on cannabinoid receptors. There are CB1 receptors that are primarily expressed in the human brain, and CB2, which are primarily expressed on immune cells. Ligands for these receptors include three major categories: the phytocannabinoids, phyto meaning plant, so that would be those found in cannabis and some other plants; synthetic cannabinoids, those that are manufactured artificially; and the endocannabinoids. These are the cannabinoid and cannabinoid-like substances that are produced naturally in the body by animals, including humans. There are over 110 different ones that have been isolated from the plant, which seem to have different effects depending on which one you look at on mood, cognition, memory, pain perception, inflammatory processes and immune response, neuronal excitability, intraocular pressure, and many other body systems and responses. The most notable cannabinoids, of course, are the phytocannabinoids, THC, tetrahydrocannabinol, which is the primary psychoactive compound in cannabis, and also the non-psychoactive class of compounds, the cannabinoids. So the synthetic cannabinoids, looking at each of these in a little bit more detail, encompass both the classical cannabinoids that are structurally related to THC, and also the cannabimimetics, including the aminoalkandols, which were developed in the early 1990s as potential non-steroidal anti-inflammatory agents. We also have the 1,5-diaryl pyrazoles, which form actually the core structure of Celebrex and some of the COX-2 inhibitors. The quinolones are used in the manufacture of many dyes and chemicals, and also are found in antimalarial drugs such as quinine and chloroquine. And the aryl sulfonamides, many of these have NSAID and analgesic properties via the voltage-gated sodium channel. So these synthetic cannabinoids have actually, their core structures have been used in approved products now for many years, but I think most people didn't realize where they actually came from. The eicosanoids are an interesting class that function in a lot of different physiologic systems and pathological processes, including inflammation, allergy, fever, pain perception, cell growth, blood flow. You can read that, I suppose, as well as I, but examples include prostaglandins, thromboxanes, the leukotrienes, lipoxins, revolvins, antioxidants. So cannabis, as you know, is regulated in the U.S., and we have to go back to 1970, which was, goodness, almost 50 years ago now, where the Controlled Substances Act was established. And that established U.S. federal policy under which manufacture, importation, possession, use, and distributions of certain substances were regulated. The legislation created, as I think most of you who are prescribers are familiar, 5 different schedules based on potential for abuse, accepted medicinal use, safety and potential for addiction. Is the drug known to be safe, for example? How likely is it to cause addiction? What types of addiction and how severe and impactful to the patient might they be? The 5 different classifications, Schedule 1 through 5, are shown here. Schedule 1 are those drugs, and sometimes these are called Schedule 1, and sometimes you'll hear it called Class 1. They essentially mean the same thing. But Class 1 drugs or Schedule 1 drugs are those for which the drug is considered, and I emphasize considered, unsafe to use, even under medical supervision. And again, as I alluded to earlier, unfortunately, sometimes political considerations have as much to do with scheduling as scientific and medical considerations. But examples, Schedule 1 drugs, aside from marijuana and cannabis, would include things like heroin and LSD. The Schedule 2 drugs, I think most are familiar. These are the products that have things like the opioids, hydrocodone, oxycodone, other things such as cocaine, methamphetamine, amphetamine, Demerol, would be other examples. Schedule 3, which would be considered a little bit less likely to be abused, would be products containing less than 90 milligrams of codeine per dosage unit. So these are the combination products, which are a little bit harder to abuse, although people do find ways. Ketamine is a Schedule 3, and interestingly, anabolic steroids and testosterone is also considered a Schedule 3 drug. Schedule 4 we're most familiar with the benzodiazepines. They're all Schedule 4. And Schedule 5 are the drugs that are most often used for things like antidiarrheal agents, antitussives, and some analgesics. Examples would be Lomotil, Lyrica, Robitussin AC. So 4 years after the Act was created, an agency to enforce the Act was created. This is 1974, when the Drug Enforcement Administration, or DEA, was established to be the lead agency for enforcement of the Controlled Substances Act. And as a Schedule 1 controlled substance, which marijuana or cannabis still is considered to be, with some exceptions we're going to talk about in a moment, marijuana use in a clinical trial still requires special licensure from the DEA, and there are registration requirements for the investigator and the site where the study is to be conducted. You'll notice where it says for more information, see 21 CFR, etc. We have in this presentation hyperlinks that will go straight to these references, but also at the end we have a reference list that has more details on where you can find more information on each of these. Because of the registration requirements, as much as the FDA actually wants to be supportive and proactive and would really like to see more investigation into the cannabinoid products, and different government agencies are indeed trying to be helpful, there are still significant delays to the clinical program to get these licenses, at least if you don't plan ahead. So if you know ahead of time that there could be a several month delay in getting these licenses, you start early and perhaps the delay would be less consequential. So things actually changed a lot just the end of last year. Many of you may have heard of the Agriculture Improvement Act, which did a lot of things actually, but one of the things it did, which I think was major and I would say long overdue, is it had to do with the production and marketing of hemp, which was defined as cannabis and derivatives with extremely low, less than 0.3% on a dry weight basis, of concentrations of THC, which is the psychoactive compound. So these changes included removing hemp from the Controlled Substances Act, because before this was passed, you couldn't even make and sell hemp rope because that was considered, a dangerous product. So hemp rope is no longer regulated, nor is anything, as mentioned, with extremely low concentrations of THC. So there are pathways now available for those who want to lawfully introduce these products into interstate commerce. And FDA is, as I mentioned, trying to be helpful to people who want to investigate these products and or sell these products in a safe and licensed way. Just want to show you a little bit of what this act looks like, because you hear something might take an act of Congress. So here's your act of Congress. And at the top, you see that the short title is the Agriculture Improvement Act. But nothing in Congress is simple. So I thought it was interesting just to look through this act, and it actually deals with, the bottom section is section 12,102. There are even more, But in this act, it talks about skim milk prices, feral swine eradication, Buy American requirements. You got to have that in the act, sheep production, and a lot of other things. So you can see how the details of this could easily get lost. But I think because of commercial implications of this, in this case, the details really weren't lost. So you can see here, which actually, hopefully you can see here. I can't. The font's a little bit small, but they define the term hemp referring to any part of the plant, including the seeds and all derivatives, etc., with a THC concentration of not more than 0.3%, as I mentioned, would fall into this act. Underneath that, then they go on to describe definition of Indian tribe. So again, an act of Congress currently has a lot of things to it. But relevant for us, of course, is the CBD or cannabinoid aspects. So as soon as this was passed, some entrepreneurial individuals immediately, this is from Amazon, started composing and marketing a lot of different products. So here you can buy your hemp oil for $99.99 an ounce, and this is superior hemp oil. If you prefer to get your hemp oil from Kentucky, it's only half that price. If you want it organic and you want it cold pressed and virgin, we're down to $13 an ounce. And if you prefer to buy in bulk, here you go, you get a gallon for, I think, $57. So some people have looked at this and obviously are making a lot of money, and they think, well, you know, this is a great way to get CBD products out there. The problem, as mentioned, is these are unregulated, so you don't know what you're getting when you're buying. There's no guarantee at all that what's on the label is what you're actually getting. And the FDA and the DEA, neither group are very pleased about this and have sent warning letters now to several dozen companies making health claims for CBD products. The FDA commissioner has stressed that CBD is a drug ingredient, potentially. It's not legal to add it to food or health products without approval from the agency. So selling unapproved products with unsubstantiated therapeutic claims is a violation of the law. Obviously can put patients at risk, and in many cases, these producers will be prosecuted. The problem is that there are so many people out there doing it now that no matter how much enforcement, most of the people are going to be able to get away with this, at least at this point. But that doesn't mean anybody's safe in doing it, and it doesn't mean that it's a wise path to take. So going back now specifically to which cannabis medications are already on the market and approved. Already 34 years ago now was the first one, and that was Marinol, also dronabinol, and this is the principal psychoactive constituent in THC that is found in cannabis. It was approved by the FDA in 1985 as safe and effective. It's been used since for HIV/AIDS-related nausea and also chemotherapy-induced nausea and vomiting. But its use is highly restricted, or at least has been, to those indications. Initially, it was put out as a Class 1 or Schedule 1 substance, but has since been increased to Schedule 2. There's talk about going to Schedule 3 because, in fact, this has not been abused very much. It was rescheduled, all right. So looks like I'm 20 years behind the time here. It's already a Class 3. We have a couple others. In 2006, Sesamet or Nabilone is a synthetic cannabinoid, which is also used as an antiemetic and for neuropathic pain. It's a, as I mentioned, synthetic cannabinoid that mimics the effect of THC. This was approved in 1985, was not commercially successful, withdrawn by Lilly, and eventually approved by Valiant in 2006. Syndrose, which is a PO liquid formulation of dronabinol, that's Schedule 2, indicated, again, for HIV/AIDS, anorexia, and for chemotherapy-induced nausea and vomiting, the same as Marinol. So things have improved even more greatly. We now have something that I think is really exciting, and that's Epidiolex, which I'm now going to pass over to Dr. Mark to tell you about that very recent and exciting approval. Dr. Mark?
So last June, 2018, FDA approved Epidiolex for the treatment of seizures associated with two rare severe forms of epilepsy, Lennox-Gastaut syndrome and Dravet syndrome, in patients 2 years and older. And as Jack may have mentioned earlier, this is the first FDA-approved product that contains a purified drug substance derived from marijuana. The previously approved products were synthetic. And it was also the first approval of a drug for Dravet syndrome. So that definitely helped the company's communications and relationship with FDA. Both are devastating syndromes. A hallmark of Dravet syndrome is that you have a typically developing child, and then all of a sudden they start having seizures, and now it's a little easier to diagnose Dravet syndrome, but historically it's been a very difficult syndrome to identify. Many of the Lennox-Gastaut syndrome patients are also typically developing, but not all, before their first seizure. The Lennox-Gastaut patients suffer from drop attacks, which put them at risk for serious injury. And then in Dravet syndrome patients, much more so than other childhood epilepsies, due to sudden unexpected death, and epileptic patients and status epilepticus are much more frequent in Dravet syndrome. So FDA reminded us at the time of the approval that advancing sound development programs that properly evaluate active ingredients contained in marijuana can lead to important medical therapies. Following the drug's approval, DEA announced that it would be placing Epidiolex in Schedule 5, the least restrictive class. Now, interestingly, during the advisory committee meeting, the controlled substances staff presented data that there is little evidence that Cannabadiol has meaningful abuse potential, even at super therapeutic doses in adults. And in the summary basis of approval, the controlled substances staff concluded that Cannabadiol could be recommended for decontrol under the Controlled Substances Act. However, there were circumstances outside the scope of the submitted studies that must be considered in determining the final scheduling recommendation. FDA also made it very clear at the time of scheduling that any non-FDA approved Cannabadiol extracts remain in Schedule 1. So here's a snippet from the drug approval package. You can see that the main difference here in what was proposed by the applicant and then recommended by FDA was the applicant proposed adjunctive treatment of seizures associated with Lennox-Gastaut syndrome or Dravet syndrome as the indication. And this is standard for anti-epileptic drugs. Adjunctive treatment, it would not be appropriate to study single AEDs in patients. And FDA ended up approving treatment of seizures associated with Lennox-Gastaut syndrome or Dravet syndrome. So even though Epidiolex will be used adjunctively, the indication approved was treatment of adjunctive seizures. The dosing regimen stayed about the same. The initial dose is 2.5 mg per kg twice daily. After a week, can go up to the maintenance dose of 5 mg per kg twice daily. And then if patients are tolerating Epidiolex and require a further reduction of their seizures, the dose can be increased to 10 mg per kg per day twice daily. 10 mg per kg twice daily. So key findings from the studies: clinically meaningful and statistically significant reductions in seizure frequency were demonstrated in 3 adequate and well-controlled trials in Lennox-Gastaut and Dravet syndromes. The most commonly observed adverse events in the controlled trials that occurred with a greater incidence in Cannabadiol-treated patients than placebo included somnolence and sedation, decreased appetite and diarrhea, transaminase elevations, and pneumonia. And generally, these events were mild to moderate in severity. Serious and or severe adverse events were generally related to transaminase elevations, somnolence and lethargy, and infections. So although no cases met Hyde's law, the risk was significant enough to require a warning and precaution for hepatocellular injury. And that warning and precaution includes monitoring of transaminases at baseline, 1, 3, and 6 months after initiation of Epidiolex, and periodically thereafter, or as clinically indicated. Now, this was not seen as a huge challenge because epileptologists are used to monitoring their patients very frequently for interactions with the different anti-epileptic drugs and keeping an eye on the seizures being appropriately medicated. Somnolence and lethargy were not surprising since Epidiolex is a CNS active drug. So it's common in the class and also common considering the polypharmacy with multiple anti-epileptic drugs. So here are the primary efficacy results. In the two Lennox-Gastaut syndrome studies, you'll see on the top 1414 and 1423, the median percent change from baseline, which is a reduction in the frequency of drop seizures, was significantly greater for both dosage groups, 10 and 20 mg per kg per day over placebo. This reduction in drop seizures was observed within 4 weeks of starting Epidiolex, and the effect remained generally consistent over the 14-week treatment period. Similarly, for the Dravet syndrome study, 1332B, the median percent change from baseline in the frequency of convulsive seizures was significantly greater for the 20 mg per kg per day Epidiolex than for placebo. And as was seen in the Lennox-Gastaut syndrome study, a reduction was seen within the first 4 weeks of treatment and remained generally consistent over the 14 weeks. That same efficacy data is presented here in figures, displaying the percentage of patients on the y-axis by the category of reduction from baseline on the x-axis. And since these studies are both Lennox-Gastaut syndrome, the seizures are drop seizure frequency during 28 days. Obviously, Epidiolex was approved, but the Division of Neurology convened the Peripheral and Central Nervous System Drugs Advisory Committee meeting to advise whether the benefit-risk profile of Cannabadiol is favorable for the treatment of seizures associated with Lennox-Gastaut syndrome and Dravet syndrome. The committee unanimously agreed that the benefit-risk profile was favorable. The committee members also agreed that efficacy was well demonstrated and that the safety concerns could be managed with labeling, education, and monitoring. A few weeks later, FDA concluded that it's clear that many patients will develop Cannabadiol-induced adverse reactions. However, those observed in the development program would be expected to be detectable by patients and caregivers, self-limited and reversible. There was no evidence within the development program that Cannabadiol causes actual harm, such as irreversible or consequential damage, although it does remain possible that there will be rare, severe hepatotoxicity once the drug is marketed, hence the warning and precaution. In general, FDA said patients and caregivers will be able to make individual decisions with respect to the drug's value to them based on the change in seizure frequency and the perceived adverse reactions. As Jack mentioned earlier, many companies are involved in developing cannabinoids for over 100 indications, including those listed here: autism spectrum disorders, traumatic brain injury, dementia, glioblastoma, hepatobiliary disorders, mental illness, multiple sclerosis, Parkinson's, Rett, and others. And as of last month, 33 states plus Washington, D.C. allow marijuana for a variety of medical purposes. So you can see there's an intense interest within regulated products and outside of regulated products.
Well, thank you, Catherine. Now, this is Scott Burian, and I'll be talking to you today about some of the chemistry manufacturing controls issues associated with cannabinoid products. Specifically, product quality standards associated with investigational new drug applications, challenges of working with botanical raw materials, and we'll kind of finish up with where does one go about obtaining the clinical trial material to conduct your clinical trial. So just for those who might not be aware and might be new to the clinical development arena, to begin the clinical portion of your drug development process, sponsors need to first file an investigational new drug application, or IND. This process provides a pathway that includes regular interactions with FDA, typically beginning with a pre-IND meeting, to support both an efficient drug development program while at the same time ensuring that patients are protected during your trial. Now, the general content of an IND is provided in 21 CFR 312 and a variety of other guidance documents that are freely available on the FDA's website. I'm not going to go into those today. We'll specifically talk about those elements of an IND that are related to product quality, specifically raw materials, your drug substance, so that active moiety that provides the pharmacologic action, the drug product, which is a formulation containing the drug substance plus other excipients to provide a deliverable product. And then obviously placebo, as they are drug products too, and will have their own quality standards. Now, what are some of the challenges of botanical products? In contrast to most small molecule drugs on the market today, which are manufactured using traditional synthetic chemistry, and they're starting out from well-defined starting materials, botanical products, on the other hand, they're derived from vegetable matter, and this can be a variety of forms, plant materials, algae, fungi, combinations thereof. So your starting material can be quite varied. They're usually prepared as complex mixtures, and you might not always know exactly what are those constituents that are providing what you think are providing the efficacy of your product. There's also batch-to-batch consistency problems. A lot of botanicals, they're grown outside. You have different environments, soils, sunlight, rain. You can get cross-pollination, obviously, and once cross-pollination occurs, you can change the genetic composition of your plants and then subsequently have potential changes in the active components derived therefrom. And obviously, again, if you're cultivating outside, there's a variety of contaminants, various microorganisms, pesticides, and heavy metals, potentially, that can be concentrated by the plants from the surrounding soil. Now, there are a number of guidance documents from FDA and additional resources in the United States Pharmacopoeia that can be of assistance when putting together the quality standards for a botanical product. First is the FDA's guidance on botanical drug development, which includes information on how to define a botanical product, and we'll talk about that specifically in regards to Epidiolex. Chemistry and manufacturing controls information, and also non-clinical and clinical considerations that need to be taken into account when studying botanical products. In addition, the USP has been updated over the last several years with a number of new chapters dedicated to the characterization and analysis of botanical products. So if you're in the beginning stages of developing a botanical drug, I highly implore you to become familiar with these resources. So what is and what is not a botanical product? As I mentioned before, the FDA's botanical guidance defines what is a botanical product and also what is not. And what is not included are any highly purified substances that are derived from a natural source. So in the case of Epidiolex, even though it is coming from a natural source, it has been processed efficiently, that it is not considered a botanical at this point. And this came up during the NDA review, and you can go to the summary basis of approval on the FDA's website for this. It's freely available, and you can read the reviewers' comments and how they went through and determined that it did not meet the definition in the botanical guidance. The botanical review team did come in and provide a consult on the botanical raw material, which Epidiolex is derived from, and commented on the quality control processes that were in place to ensure consistent quality of that raw material. So if you are developing a highly purified drug substance, maybe like Epidiolex, and using a botanical raw material, you want to make sure you have sufficient control over where this material is coming from. So you want to be able to provide the name and address of the growers, description of the plant species that you're using, harvest location, and in the reviews that I've seen, they're even asking for GPS coordinates. So they want to know exactly where this materials are coming from. You want to indicate where the plants are harvested, the growth conditions that are associated with the time that you've harvested that material. Describe how the materials are handled post-harvesting. Are they washed, dried? Do they go through any mechanical separation or maybe size reduction procedures? How are foreign materials controlled? Bugs, non-plant material, or maybe parts of the plant that aren't active and you want to remove as you further process the material. If the materials are preserved, you also want to describe that. And then how they are transported and stored before being further processed into the final drug substance. So when you're setting up your supply chain, you want to make sure that you're developing the appropriate quality standards for your botanical raw material. So as I mentioned earlier, plants are known to concentrate heavy metals that might be in the surrounding soil, so you want to make sure that you're implementing an elemental impurity test and setting appropriate limits. Microbial contamination needs to be monitored, especially for fungi, which could produce, in the case of aspergillus, aflatoxins, which are known to be a carcinogen. And then typically when growing outdoors, it's not uncommon to use pesticides to reduce crop loss due to insect infestations. However, those chemicals, while beneficial, can be retained on the plant matter you're harvesting and then ultimately wind up in your final product. So since the pesticide is not supposed to be there, it would be considered an impurity, and you would need an appropriate method and specification to control for these. And finally, you don't want to have any other foreign materials or adulterants in your raw material that could be passed along to the final product. So now that we've covered some of the regulations concerning INDs and quality standards for botanical-based products, let's talk a little bit about how one might go about getting the raw materials for a clinical investigation involving cannabis. So in contrast to what you might think, given the availability of cannabis at dispensaries in a variety of states that have legalized cannabis for medicinal and recreational use, the FDA considers the National Institute of Drug Abuse, or NIDA's, drug supply program as the only legal source of cannabis in the U.S. So for nearly 50 years, the United States has relied on a single grower to produce marijuana use for medicinal research. Under this arrangement, NIDA has been contracting with the University of Mississippi's National Center for National Products Research to grow and process marijuana under the appropriate DEA registrations. So what can you get from the University of Mississippi? Well, they manufacture a variety of strains upon request by NIDA and are capable of producing a variety of end products, including bulk plant material, marijuana cigarettes, cannabis extracts, and purified cannabinoids. Now, I spoke earlier about the quality information that you will need to supply in your IND for a botanical raw material and drug substance. And so to assist researchers in providing the appropriate information in your IND, the NCNPR has 2 active drug master files on file at FDA for sponsors to reference. Now, for those of you who have never referenced a drug master file, these are documents that contain proprietary information from a supplier, but they're in a format that allows the FDA to review that information when they're reviewing your IND, but keep it from any sponsors themselves. So a sponsor never actually looks at the drug master file, but you are provided a letter of authorization, which you would include in your IND that tells FDA you have authorization to reference the information in the master file. Now, if you look at the NIDA Drug Supply Program catalog, again, freely available on their website, you can see the material that they're providing for research. Variety of plant materials and also pre-made marijuana cigarettes contain varying amounts of THC or CBD. However, if you are conducting a study, perhaps you want to be using the NIDA supply marijuana cigarettes, the Drug Supply Program catalog does have a number of caveats for that product. They clearly state that their supply is limited. They're not planning on manufacturing any new stock in the near future. And if you do obtain materials from this program, you want to, A, confirm of their availability, and you might also want to have the material assayed for THC or CBD content, depending on whatever you're researching, before you begin your clinical study. Now, it should be noted that the catalog was last updated in 2016. So hopefully, with the recent approval of Epidiolex and the renewed interest in studying the effects of THC and CBD compounds, that NIDA might have some impetus to revise the products being offered in this catalog so that there's better consistency between the materials available to sponsors and also what is available at dispensaries in those states that allow such products. Because if you look at the concentrations of THC and CBD in the NIDA catalog, and if you were to look at what is available in states where it's been legalized, you'll see that there's quite a bit of difference. And so depending on what you're trying to emulate, you want to make sure that you're using similar materials. Now, the NIDA program at the University of Mississippi was originally designed to supply marijuana for federally funded research and not the commercial research market. So And until just a few years ago, there was really no clear legal pathway for entities outside the NIDA system to supply the commercial pharmaceutical market. But as a result of the demand in cannabis products for medical research, the DEA, in consultation with NIDA and FDA, has published in 2016 a new policy to increase the number of entities registered under the Controlled Substance Act to grow marijuana to supply legitimate researchers in the U.S. Registrants can apply for their registration and manufacture marijuana by filling out DEA Form 225, which is available on the DEA website. But in reading the law blogs about this, the DEA was quoted as cautioning that they only intend to increase the number of registered cultivators for supplying researchers to produce an adequate and uninterrupted supply of these substances under adequately competitive conditions for legitimate medical, scientific research and industrial purposes. So this is not going to be the floodgates opening and a great land rush for people to come in and start growing marijuana. The DEA appears to be keeping a tight lid on their new pathway to becoming a grower. And if you are a grower and you are considering filing for a registration to manufacture a Schedule I substance, you need to be aware of the criteria that the Controlled Substance Act, the DEA will follow and consider when they're evaluating your application. And one of the major items that the DEA said they were going to place emphasis on is Factor 4, and that is whether the applicant has engaged in a legal activity involving controlled substances. Now, in this context, the DEA has stated that legal activity includes any activity in violation of the Controlled Substances Act, regardless of whether such activity is permissible under state law. So you might be in a state and growing and servicing the medical marijuana market in your state. That is technically violating federal law. So even though you might have the know-how and the conditions and the facilities to cultivate marijuana, in the eyes of the federal government, you are violating the Controlled Substance Act. And the DEA has noted that while past illegal conduct does not automatically disqualify an applicant, it may weigh heavily against granting the registration. So you need to be careful when you're approaching this and how you might want to get into this industry.
All right. Thanks, Scott. I'm going to shift gears and talk just a little bit about non-clinical safety evaluation of cannabis-derived therapeutics, some considerations for abuse liability assessment, and then some regulatory considerations, special programs that are available for drugs being developed for severe or life-threatening illnesses. So, operator error here. All right, so for small molecules, as Scott had indicated, those are either synthetically derived or they're highly purified extracts containing one or a couple active molecules. And those would, the non-clinical safety evaluation, those would be very similar to other therapeutics. They're small molecules and they would follow, if they're a new molecular entity, they would follow 505(b)(1) regulatory pathway, and they would, an ICH-M3, which is a guidance, International Conference of Harmonization Guidance, M3, which describes non-clinical safety studies that were required for conducting human clinical trials and also for marketing authorization. And so these molecules would need to be evaluated by general tox studies, reproductive and developmental toxicology studies, genetic toxicology studies, and if it's a chronic indication for carcinogenicity. In addition, safety pharmacology would be expected as well, evaluating effects on the CNS, respiratory, and cardiovascular system. For drugs that are small molecules that are repurposed, such as Syndros, which is a new formulation of Marinol, and the same route of administration, or if it's even a different route of administration, they can follow the 505(b)(2) guidance for non-clinical safety evaluation of reformulated drug products or products intended for administration by an alternate route. For extracts, complex mixtures, as Scott had indicated those are less purified and may contain multiple components that contribute to the activity, some of which may not really understood all the molecules that are contributing. Those would fall under the botanical guidance, and there are some information in the botanical drug development guidance that pertains specifically to non-clinical development. The one important consideration here when we think about non-clinical development of botanicals in the case of cannabinoid-derived therapeutics is that there are no lawfully marketed dietary or food supplement cannabinoid-derived botanical products. Therefore, relying on the literature may or may not be may or may not provide enough compelling information to support safety. And it's still expected that a full evaluation of the safety and of the product to support initial clinical trials is going to be required. And so that's going to require, that is going to require general toxicology studies, safety pharmacology studies, genotoxicity studies, and possibly even carcinogenicity studies, as well as pharmacokinetics. And here it can be more challenging for pharmacokinetics because you have multiple, you have a complex mixture. And so evaluating the exposure as possible for chemical constituents that could contribute to the toxicity and or pharmacology is going to be expected. Metabolic states of these chemical constituents should also be evaluated as possible. Reproductive and developmental toxicology studies will be expected, and if you're conducting clinical trials in pediatrics, juvenile toxicity studies will be required to support that. And it will be expected that you'll summarize all the available non-clinical and clinical literature with a critical review of the quality of the data and its relevance to your program. So I'm going to switch gears and talk a little bit about these liability studies. These form the basis for the risk assessment that goes into determining the scheduling, as Jack discussed earlier. There is a guidance for assessing abuse potential of drugs, and one of the important features is that if you have a brain-penetrant molecule and it's CNS active, then abuse-related animal behavioral studies are required. Such as these studies include drug discrimination studies. Does the drug produce effects that are similar to known drugs of abuse? And self-administration studies. Does the drug have rewarding or reinforcing behavior? In addition to physical dependence studies, after chronic administration, are there withdrawal signs upon abrupt drug discontinuation? In addition, you'll be looking at some of your safety pharmacology studies, and you'll be looking at whether there are any abuse-related signals in your safety pharmacology or toxicology studies, such as hyperactivity indicative of stimulation, somnolence or sedation types of behaviors. And you'll be looking at that as well as the comprehensive receptor ligand binding studies that will need to be conducted, including receptors that are known to be related to abuse potential, and several of those receptors are actually listed out in that FDA guidance. PK studies are of course needed because and it's important that when you're running these abuse liability studies non-clinically, for several of them, that you take into consideration that recreational drug users typically use drugs that doses that are multiples of the medically recommended dose. And so that needs to be taken into consideration when you're considering doses for your studies. And it's important to discuss these animal study designs at your end of phase 2 FDA meeting for your abuse liability non-clinical studies. So The information from your non-clinical studies and from some of your clinical programs, we're going to go into determining whether human abuse potential studies are going to be needed. And if the responses in animals and general behavioral studies are similar to those of known drugs of abuse, and some of the other rewarding properties that support animal self-administration, and what are the profile of the abuse, are there any abuse-related AEs, such as euphoria-related AEs in your clinical studies? And that information is going to help determine whether or not abuse potential studies, specific abuse potential studies are needed. It is likely that they will be needed, especially for novel cannabis-derived therapeutics. Okay, so, and then a risk assessment, once you have compiled all of your abuse data, you're going to put together a risk assessment, and the risk assessment would take into consideration the abuse-related studies in animals and in humans, abuse-related AEs from your clinical studies, whether or not there is any information related to the overdose, whether intentional or accidental during the clinical studies, your human abuse potential study, and all of this will be summarized into a risk assessment with a proposal for scheduling and product labeling. So here's just a couple of examples. Epidiolex, what went in from a non-clinical perspective, they did do assessments. They found that they had no binding to any of the cannabinoid receptors or other receptors associated with drugs of abuse. In addition, there were no overt behaviors that were similar to those produced by drugs of abuse. There were no cannabinoid agonist response in an abuse liability test, and it did not generalize to Dronabinol or midazolam, which is depressant in drug discrimination studies, showing that it does not produce effects similar to a cannabinoid agonist or benzodiazepine. And it did not produce self-administration, suggesting it didn't have any rewarding properties or properties that are known for drugs of abuse. In addition, in their clinical studies, they had no euphoria-related adverse events in their phase 1 studies. It wasn't evaluated in phase 2 or phase 3 because of concomitant medications that would confound the interpretation of the data. Overall, their assessment was from preclinical and clinical studies that Epidiolex has negligible abuse potential, and it was deemed, based on this, to be Schedule 5. Now, Marinol was developed and approved prior to the FDA guidance, but there were, early on, this is the synthetic THC, that there were some indications from non-clinical studies. There were indications of tolerance, and initially you saw some CNS depression, but then after tolerance, which happens after 3 days of treatment, there was stimulation of the CNS stimulation activities. And in clinical studies, there were easy laughing, elation, heightened awareness, in 24% of the Marinol subjects. They also had tachyphylaxis and tolerance as well, and they had an abstinence syndrome, which is, they showed physiological signs of withdrawal after abruptly ceasing the drugs. This was deemed to be Schedule 3 because addiction was uncommon. There was no evidence of decreased motivation, cognition, judgment, or perception, and both psychological and physiological dependence was observed in healthy individuals, but addiction was considered to be uncommon in the patient population, what it was indicated for. Syndrome, on the other hand, they did also conduct an abuse potential study that showed that the abuse potential for syndrome was very similar to the Marinol capsules, and syndrome is essentially an oral solution similar to the Marinol capsules. The controlled substance staff was consulted. They had initially proposed Schedule 4 for their drug, and the controlled substance staff at FDA determined that Syndrome had greater potential for abuse than the Marinol capsules and other Schedule 3 drugs, and it presented a higher risk of unintentional overdose if abused, and so therefore they gave it a Schedule 2. And lastly, I'm just going to briefly switch gears again to talk a little bit about some of the benefits if you are Epidiolex if you are developing drugs for severe or life-threatening illnesses such as Epidiolex. And here, this just really demonstrates FDA is invested in getting drugs to the market that meet safety and efficacy criteria for severe and life-threatening illnesses, particularly as they provide compelling evidence of benefit versus risk to the patients with respect to alternatives or there are no alternatives. Epidiolex had orphan drug designation. They have fast track designation, priority review, rolling review, pediatric disease voucher, which is quite valuable. They had an expanded access program. And here's the FDA guidance where you can read more about particular programs for developing drugs to meet unmet medical needs for severe and life-threatening illnesses. And I'm going to stop at that, and we can take some questions.
Okay, one of the questions
Okay,
we just had.
we're ready to move on to Q&A. Just a reminder to the audience, you can submit questions using the Q&A button at the bottom of your screen. We have a lot of great questions already. We'll try to get to as many as we can. So this first question, I'm just going to toss out to the panel. And the audience member says, if the clinical study is not conducted on U.S. soil, do we still need Schedule I license from the DEA? And could this be a strategy to conduct trials outside of the U.S.?
I would have that conversation with DEA first. In order for the phase 3 data to be, kind of respected by FDA as applicable to a U.S. audience, you wouldn't want to conduct all of your studies outside of the U.S. But yes, it should not be required to have a DEA Schedule I license outside of the U.S.
And this is Scott, but obviously you'll need to abide by whatever controlled substance laws are in force in the jurisdiction that you're planning on running your clinical trials. So.
And import-export will still be a significant matter to deal with inside the U.S.
Yeah.
Anything coming in and going.
Right.
Okay, thank you. And we have another question that came through that I just want to take real quickly because this has been asked several times, and that is that the CBD products are now hitting the shelves of CVS, Walgreens, some of the grocery stores, and in fact, we just learned that there's a major hamburger chain that's putting it in the hamburgers. And the question is this legal? Well, we're not lawyers, but I can tell you a couple of fundamentals. One, if any of those have 0.3% or higher of THC, no, it's not. Now, probably all of them claim that they're less than 0.3, but upon testing, some aren't. And when that's discovered, there are going to be a lot of people in a lot of trouble. The other thing that is not legal is for any claim to be made, any health benefit or anything that's even an implied benefit for a specific indication. And a lot of these manufacturers are doing that. The question comes up, what about the chains, CVS, Walmart, whoever it may be, who are actually selling this? Do they have some liability? That I have to leave to the lawyers. I'm sure there'll be much more said about that.
In developing Epidiolex, we heard from epileptologists after epileptologists that they were very concerned for their patients who were being administered over-the-counter CBD. Because the batch-to-batch variability is just unknown, and patients who are having seizures can't tolerate that.
And that's one of the biggest harms. We often think about, okay, they might take something that might have a little too much THC or some contaminant and have direct harm, but there's the indirect harm of people with potentially serious illnesses who are taking unregulated products that either don't work at all or may make things worse.
Right, and the FD&C Farm Act in December of 2018 THC and CBD drug products, if they are above that threshold, are excluded from the dietary supplement definition, and they cannot be sold as dietary supplements. They're excluded on the basis that they have already been authorized for investigation as new drugs for which substantial clinical investigations have been instituted. So if there was an IND or an open IND and clinical studies, then the cannabis-derived therapeutics, they will be excluded as being dietary supplements, and that is FDA concluded that this is the case for THC and CBD, so they cannot be developed as dietary supplements.
Unfortunately, we've run out of time for our Q&A session. We had a lot of great questions today and couldn't get to them all, but we will be trying our best to get back to everyone who submitted personally after the webinar. I'd like to thank everyone for attending this Fierce Markets webinar and submitting so many of those great questions. I'd also like to thank our speakers for participating and Roe for presenting today's webinar. As a reminder to the audience, this webinar has been recorded, and you will be able to access the recording within 24 hours by using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.